My name is Ralf Schmid. I'm a researcher at the University of Pennsylvania. I work here at the gene therapy program, and my group is primarily interested in rare neurological diseases. In the following slides, I will give you a few talking points on gene therapy and viral-based approaches. Which is directly complementing what you just heard about using small molecules to try to cure diseases. So why are we interested in anything with the word gene in it? Well, one of the reasons is that the vast majority of rare diseases, about 80%, are genetic in origin. So we really have an opportunity here once the genetic defect has identified trying to directly alter the genetic disposition of the patient, where small molecules typically just trying to alleviate symptoms and kind of curing around the genetic defect. So how do genes work? And I just want to take a step back that you all understand the process. Normally we start with the gene is encoded by DNA. That gives rise to an intermediate form, which we call RNA, and then eventually we end up with a protein. The protein travels to where it has to be in the cell and fulfills its function. So now if there's a mutation in DNA, a pathogenic mutation, that precludes the DNA from turning into RNA, and then there's also no protein, or the protein is so short-lived that there's essentially none there. And that is true for a lot of the genetically encoded diseases. So now the opportunity then basically is to trying to repair this right on the genetic level. So the one thing what you can do is gene therapy. So you basically add the gene, you add an additional gene, the same gene which is already there with the mutation, but you just add the fully functional one. You add DNA for that. Or you use something which is called gene editing or CRISPR, as you heard before, which are tools to basically revert this mutation and turn the mutation into the normal genetic code. Both methodologies then basically would allow to produce RNA, then we get protein, and so then the cell can resume its normal function. So now, gene therapy and gene editing, both methodologies consist of really two components. So the one thing is our delivery vehicle, and that could be something we see all the time, which has been established in research already. They like the FedEx vehicles or planes, you know all about. But then this could be also something which is emerging or new. FedEx analogy would be a drone or a robot. And so this one, and I will expand on that a little bit more in the following slide, the delivery vehicle is typically virally derived, or it is some chemical mechanism how to get this piece of DNA or the gene editing machinery actually into the organism. And then the other important component, of course, is the payload. And so the payload now, that those are the packages being transported by the FedEx trucks, they can be of various shapes and sizes. So what that allows to do is really you can do a direct modification, a gene replacement. So one of those boxes would have the gene in, gets put in the body, and it ends up in every single cell and is starting to make the protein which was lost. The indirect approach would be that this package includes instructions how to regulate gene activation. So that could help to either switching a gene on, if that is helpful for the disease. If, for example, in some diseases, you still have a normal copy of the gene left, so you could switch that on to overcome the disease symptoms. Or in other cases, it might be that there is a mutant expressing, and by simply getting rid of that mutant gene, mutant protein, you can improve the disease symptoms. And you can deliver those regulators, and they're highly specific regulators. We can just target the gene of interest. And then the other indirect method, of course, would be the editing. You can edit on both levels. You can either edit the DNA itself, that's the human genetic code, that's kind of a one-time and done thing, so that's a permanent change to the human genome. Or you can edit the RNA, that's intermediate before the protein is made, that is not permanent. So you're not touching the genetic code, you're just basically repairing what comes out of the genetic code. So why is anything with gene therapy, gene editing, really, why might that be game changing, and why there is so much buzz about it? Well, what we think that those technologies can really provide a single and durable administration, one-time administration. And I think one of a good example is SMA gene therapy, Zolgensma, which was approved a few years ago. So right now, some of those patients are in 6 years post gene therapy. So they were giving gene therapy to repair the genetic defect, and then the condition kept improving, and the children are still very mobile and have overcome a lot of the disease symptoms 6 years after a one-time treatment. Of course, time will tell how long really this single durable administration will hold, if it's really decades, or if, let's say, after 10, 20 years, you need to do some kind of re-administration. So now let's talk about the different modalities, how you can actually administer your gene therapy or gene editing. The one option is viral gene therapy. So what you typically would be using is just the shells of a virus, also called capsids, and I have a picture, computer animated picture on the left side here, which can provide really efficient delivery. And I know, looking at that, you probably say, oh, well, this just looks like COVID, which is true because it is a virus. But in this case, we would only use the outside, not any of the payload, which is inside that virus. Because given for COVID, for example, it is really what's inside that virus what makes people sick. So the technology is throw all of this away, and basically we just rely on delivery vehicle, which is the outside. So for human therapeutics, we mostly rely on lentivirus or adeno-associated virus or AAV. Lentivirus, it integrates permanently in the genome, so it just adds itself basically to the human genetic code. So it's really used a lot for ex vivo manipulations, where you would take patient cells out, you modify them, and then you do a lot of quality control steps before you re-implant into humans to make sure that everything is okay, since it's a permanent integration. AAV and on the other side is the AAV genome, the payload, persists separately from the human genome in the cell. So you're basically just adding an additional piece of DNA, which is also functional inside the cell. And that's why this is really the workhorse now for in vivo applications, where you would directly inject the AAV loaded with a gene therapy or with the gene editing tools into the patient. Another modality is adenovirus. You might heard about that. That's the basis really for the J&J vaccine delivering COVID spike genes. It is very immunogenic, so that's why it's really good if you want to cause an immune response and train the immune system. But it is less optimal if you want to cure disease, which has nothing to do with the immune system. The non-viral or chemical gene therapies, which can be used, those are for the most part liquid nanoparticles or LNPs. That is really an emerging technology, but I think it really leapfrogged into the public view, and we learned a lot about it because that is the basis of the Pfizer Moderna COVID vaccines. And so LNPs are basically another way to deliver genetic information, either DNA or RNA, to the patient. Then the question is, of course, well, replace a broken gene via gene therapy or just fix it via gene editing, and that really depends on the context. what you need to achieve. If you really need to replace something, if you need to down regulate your gene, so what your target organs are, if you have one target organ, if you have multiple target organs, your patient population, if there's one prevalent mutation, and basically targeting that, you would help to cure a large set of patients, or if it's really a lot of different mutations, and so rather replacing the gene. And of course, I mean, it's what is the safer way? What is the safer way in the end to overcome a genetic disease? Okay, so real quick, some routes of administration. So you can either apply AAV technology intravenously, just injecting into the bloodstream. You can directly inject it. For CNS, it would be in the brain for the most part. All the other organs are easier reachable for IV. Or you can go for CNS indications, you can go through the CSF, the cerebrospinal fluid, which is either intrathecally, where it's administered by a needle in the spinal canal, or by injection directly in what's called the cisterna magna, which is closer to the brain, as a fluid-filled cavity. Those three modalities on the right are really the good choices right now for any CNS indications, because unfortunately with IV you can reach about any organ except for the brain, because there's something called the blood-brain barrier, which really precludes anything from seeping out of the blood and entering the brain tissue in an efficient way. So people have been trying to address this for a while now, trying to find AAV, specialized engineered AAV, which could do just that, because AAVs are normally kept hindered by the blood-brain barrier to cross over. It has worked really well in mice. So if you inject mice with green fluorescence protein DNA encapsulated in AAV, the brain lights up vaguely. Unfortunately, researchers still working on translating this kind of spectacular approach into that it works also in primates. So that's why really, I mean, delivering to the spinal cord, into the CSF, it's very efficient. Neuromuscular disorders can be addressed that way. Direct injections into the brain or any other nerve tissue, really it has to be a specific cell population. It's highly local, for example, like for Parkinson's disease. Global brain delivery of AAV gene therapy to the brain is at the current point somewhat inefficient. It works really best if you have something which is called a bystander effect, where the cells which receive your gene therapy then can secrete the protein after it's made, and so repairing all the surrounding cells as well. But that's obviously a highly specialized situation where you're dealing with secreted proteins.